Shaped structures are crucial. Worker bees are conducted under sterile grading procedure is critical.
Altering natural component proceeding to the packaging fee across all trays.
The process is precisely
The film surface, detect
quality royal jelly, ensuring optimal conditions for the colony.
work this. In modern beekeeping, the formation of queen cells is meticulously monitored. These teardrop-shaped structures are crucial. Worker bees nourish the developing larvae with specialized nutrient-rich secretions, ensuring the growth of the next generation.
To maintain peak nutritional density, our raw material selection standards mandate a pure.
Structural uniformity observed here is critical for industrial-scale production, ensuring maximum efficiency and resource allocation across the colony. Our advanced climate control systems maintain the optimal temperature, which is crucial for the synthesis of high-quality royal jelly.
Scrubbers. Worker bees utilize a specialized nutrient solution, royal jelly, secreted from glands in their heads.
This royal jelly exhibits exceptional purity and biological stability, essential for the queen's development.
Worker bees nourish the developing larvae with specialized, nutrient-rich secretions, ensuring the growth of the next generation.
We introduce the selection standards for functional food raw materials. Only ingredients with proven bioavailability and stability are approved for use. Raw material selection for the functional food industry adheres to strict guidelines, ensuring peak nutritional value and complete traceability from the hive.
We are now observing the queen cell creation phase. These uniformly hanging teardrop cells are central to our modern beekeeping process.
Biosecure harvesting chamber. To maintain absolute food safety, all procedures are conducted under sterile conditions using precision instruments.
The diet of the queen bee is synthesized by the nurse bees. It's a potent mix genes.
The nutritional composition of royal jelly is complex, featuring high levels of protein, beneficial fatty acids.
Analyzing the biological value reveals a high concentration of unique natural proteins, crucial for cellular regeneration and growth.
Pre-harvest analysis indicates high quality. The royal jelly exhibits excellent uniformity and ideal moisture content.
The secretion process involves the hypopharyngeal glands of the worker bee, creating this substance used exclusively to nourish the queen.
Secreted by the worker bees, this substance provides the exclusive nourishment required for the queen's development. Raw material value assessment finalized. Purity levels confirmed. Technological collection phase initiating now.
Welcome to the Biosecure Harvesting Chamber. Please follow all protocols for certified food-safe extraction procedures.
Adherence to standard operating procedure is critical. Maintain precise temperature and spacing to preserve royal jelly nutrients.
Adherence to precision handling standards is required to preserve all vital nutrients.
This segment details the closed-loop logistics chain. The queen cells are transported to the next processing zone for royal jelly extraction. The closed-loop logistics chain ensures maximum purity in royal jelly production. These queen cells are transported to the next processing zone.
Our advanced error reduction technology ensures precision in biological raw material harvesting, minimizing contamination risks.
The implementation of AI-driven robotics has reduced biological raw material harvesting errors by 99%.
In accordance with strict protocols, all personnel adhere to rigorous hygiene standards for functional food production.
In the functional food industry, strict hygiene standards are maintained continuously. Our synchronization ensures
The harvesting phase is now complete. All specimens are secured for subsequent processing. The purpose of the precise wax removal is to standardize the raw materials before the extraction process begins. Maintaining industrial hygiene standards is critical in biological raw material pre-processing.
automation ensures material consistency, allowing thousands of queen cells to be processed simultaneously at uniform speed.
migration. The multi-tiered system ensures that thousands of queen cells are processed with absolute precision. This commencing preparation for closed extraction.
The importance of shape standardization before fluid extraction is critical for quality control.
Preliminary cutting stage complete. Commencing preparation for closed extraction.
Through precise pressure regulation,
the natural enzymes are preserved, maintaining the highest level of purity.
The stabilized pressure readings indicate optimal conditions. This precise environmental control is crucial for preserving the
Our closed-loop system ensures a contamination-free process. This no-contact methodology is the future.
Maintaining a consistent low temperature is crucial during the extraction process to preserve the royal jelly's biological integrity and premium quality. Analysis confirms optimal production efficiency and high material uniformity across all active modules.
Phase complete. No contact extraction. Proceeding to preservation stage. No contact extraction phase complete. Commencing preservation stage.
Rapid cooling within the thermal layer is critical. This ensures the preservation of enzyme activity and the structural integrity of the royal jelly. Maintaining a stable temperature, precisely 4 degrees Celsius, is critical for preserving the integrity of the royal jelly during bioproduction.
Through rapid temperature reduction, we preserve the delicate nutrient structure of the royal jelly. This ensures maximum stability and efficacy.
To preserve the integrity of the royal jelly's biological structure, the agitators maintain a very low speed.
Automated monitoring systems confirm raw material quality. Temperature stabilization is now complete.
Pre-processing phase concluded. Stage complete. All systems nominal.
The pre-processing phase is concluded.
Centrifugal force removes microscopic particles without altering enzyme structure.
Centrifugal force removes microscopic particles from the royal jelly without altering the crucial enzyme structure.
All systems are optimal. Vibration control standards are confirmed for biological food purification.
Maintaining a stable temperature of 4 degrees Celsius is crucial during high-speed separation to preserve the integrity of the royal jelly. The system maintains a precise temperature of 4 degrees Celsius to preserve the integrity of the royal jelly during high-speed separation.
The multi-stage purification process ensures maximum potency and safety.
Is complete.
entrance. Real-time data streams confirm the stability of the biological culture. Purity levels remain high, and
Successful removal of microscopic impurities.
Consistent quality is maintained through rigorous data analysis. The purity index is now optimized. Purification is complete. All microscopic impurities have been successfully removed.
As automation increases, the human role shifts to supervision, analyzing data to maintain optimal processing parameters for the royal jelly.
The objective is a microscopic structural analysis of the royal jelly, examining cellular integrity and composition.
As indicated by the stable temperature graphs, optimal conditions are maintained.
As the royal jelly is carefully dispensed, observe the precise volume and uniform circular shape required for accurate analysis.
Operational analysis confirms level 1 stability across all royal jelly storage tanks. The functional food matrix is optimized for maximum yield.
The parameter monitoring phase is complete. Purity level standard achieved. Commencing laboratory testing. The royal jelly structure is confirmed to be uniform, with 0 impurities detected. Parameter monitoring phase complete. Proceeding to laboratory testing.
Scientific quality verification process complete. All structural data points are within acceptable parameters.
Analysis complete. Quality indicators are standard compliant. Scan result, pass.
scientific quality verification process initializing. Analyzing structural data. Verification successful.
Purification and refinement are complete. Proceeding to product stabilization.
We use sublimation principles to remove water directly from the solid to vapor phase, which preserves the nutrients in the royal jelly.
Moisture removal at sub-zero temperatures is critical to preserve the active biological enzymes. Sublimation principles remove water directly from solid to vapor, preserving essential nutrients.
Ulcers. The freeze-drying process is crucial for preserving sensitive compounds. It retains the full natural bioactivity and
precise temperature and pressure control are critical during the lyophilization process to maintain the delicate nutritional structure of the royal jelly. The Syncret system maintains precise temperature and pressure control, currently at -50 degrees Celsius.
The process of biological moisture removal is now complete. Product is ready for extraction. Complete. The process was successful. Freeze-drying technology preserves long-term quality without altering natural components. Freeze-drying technology preserves the long-term quality of the royal jelly without altering its natural components. Optimal purity is characterized by the vibrant golden color, high natural sheen, and a smooth, continuous flow without crystallization. The refinement cycle is successful. Observe the perfected physical structure, stable and uniform at 35 degrees Celsius.
Optimal purity is indicated by the pale golden color, high natural sheen, and smooth, continuous flow. Ideal viscosity confirmed after processing. Flow rate is stable and consistent. After refinement, the physical structure is perfected, ensuring maximum stability and uniformity.
All royal jelly must meet strict homogenization standards before proceeding to the packaging phase.
All batches must meet the specified homogenization standards prior to packaging. Ensure consistency across all trays.
The deep refinement process ensures exceptional structural stability, as evidenced by the perfect uniformity of the royal jelly.
Through advanced deep refinement, the structural stability of the royal jelly is maximized, resulting in this perfectly uniform, viscous texture.
The product has achieved high purity and is ready for standardization and packaging.
The homogenizer uses high-speed rotation to generate micro-shear forces, ensuring even molecular dispersion of the royal jelly.
The homogenation process is stable. Temperature is maintained precisely at 25 degrees Celsius for optimal biological integrity. Temperature control is precise, ensuring the integrity of the sample at a steady 4 degrees throughout the homogenization cycle.
Mechanical shear breaks large molecular clusters to achieve optimal smoothness. The mechanical shear breaks down large molecular clusters, resulting in this optimal smoothness and a homogeneous consistency.
Homogenization ensures uniform particle distribution, enhancing the stability and extending the shelf life of the royal jelly product. Quality assessment confirms successful homogenization. The product exhibits uniform viscosity and optimal particle size distribution.
Completion of smoothing before transfer to commercial packaging lines. Structure stabilized. Process complete. Transferring to commercial packaging lines.
The dark amber glass is crucial. It protects the royal jelly from light-induced oxidation, maintaining peak potency. Utilizing advanced automated precision dosing, we ensure the purity and exact measurement of every milliliter in our premium food production.
Utilizing non-contact packaging principles ensures absolute hygiene. This system minimizes contamination.
This high-precision system ensures an airtight seal, preserving the delicate biological activity and maximizing the lifespan of the royal jelly.
Real-time optical inspection guarantees a perfect pass rate, demonstrating the efficiency of total automation.
Transition complete. The royal jelly is now in stable commercial form, prepared for inspection and distribution. Stable commercial form, ready for inspection and distribution. The benefits of this automation are clear. We have achieved a perfect pass rate, ensuring consistency across all thousands of units.
The sealing process is critical. It prevents oxidation, thereby
The sealing process prevents oxidation, thereby preserving the biological activity of the royal jelly.
Our airtight sealing mechanisms maintain product purity throughout the modern production chain.
For preserving the natural nutrients and ensuring the long-term freshness of the royal jelly.
The synchronized servo mechanisms ensure precise adherence to industrial packaging standards.
Precision sealing achieved. Gasket compression verified at 99.9% efficiency. This final check confirms proper sealing, which ensures long-term storage stability and
Proper sealing ensures long-term storage stability, a critical factor for maintaining product integrity.
Technology creates an airtight seal, shielding the royal jelly from both light and oxygen, preserving its potency.
Biological structure. The encapsulation process is precisely monitored for optimal temperature.
Pharmaceutical grade size uniformity is maintained with 99.9% seal integrity.
Our advanced softgel technology provides a vital protective barrier for the sensitive compounds within.
The capsule shell plays a crucial protective role, shielding the sensitive active compounds housed within the gleaming softgel.
The stabilization process utilizes controlled temperature and humidity to ensure the structural integrity required for extended shelf life.
Through precise control of temperature and humidity, we achieve optimal structural stabilization, guaranteeing an extended shelf life.
The transformation from liquid royal jelly to these premium commercial capsules is executed with near-perfect compliance.
Pharmaceutical and nutraceutical production.
Traceability ensures every jar can be tracked from production to consumer, guaranteeing quality and safety standards are met. Compliance with all functional food labeling regulations.
Automatic inspection eliminates all positional deviations, ensuring flawless.
Automated quality control ensures every jar meets our stringent standards. The vision system analyzes each product at high speed. We guarantee total transparency, providing all product details up front for your assurance.
Physical impact, ensuring maximum potency.
All units are verified for authenticity and quality. This is the golden standard of
packaging standards for transit. The shrink wrap application ensures a moisture and dust-proof seal.
The temperature is precisely monitored at 200 degrees Celsius, ensuring a flawless seal and complete food safety.
9002 quality standard before entering the shrink wrap sterilization zone.
The inspection camera system scans the film surface, detecting imperfections before transfer to carton packing.
To ensure the long-term efficacy of royal jelly.
All systems operational. Temperature stabilized at -20 degrees Celsius. Humidity at 65%.
Our intelligent warehouse management system ensures consistent product quality.
Our technology provides real-time temperature tracking for optimal preservation and integrity throughout the cold chain. Our automation protocols are designed to minimize human error, ensuring the integrity of the food logistics supply chain. The cold chain is maintained as the shipment transitions into the distribution network.
The automated loading sequence is complete. The royal jelly is secure and temperature stabilized for transit.